Then share the screen. So in the last class, we were talking about gene therapy, and gene therapy is to correct the errors in the DNA. And DNA is a template that really regulates our life. And this DNA is made of 4 alphabets, ATGC. And that 4 alphabets is enough to control our life. It has all the programmings that unfolds right from the stage we were as a zygote, right? As a zygote, we were a single cell. And from there, our life unfolds. And first 9 months, when we are inside our mother's body, there's a clock running, and that clock has to run its course in exact time and exact seconds or nanoseconds. If there is any delay, then can be problem with the baby. So, but there are situations where there are errors in the DNA sequence, and that error leads to very serious diseases. And so that cannot be treated like headache or fever or vaccines. Those are permanent, and that causes a lot of emotional pain, and to the person, to the family, to the community that undergoes or experience those kind of genetic abnormalities. Um, so what's the solution for that? And the solution was to change or edit the DNA, right? So in the last class, we talked about various vector-based system. So these vector-based system, gene is transfected or transduced into this disease cells, or could be bone marrow. What scientists are looking for to make a permanent change. Permanent change is to change the DNA sequence itself using some tool. In the last class I mentioned to you, we can use lentivirus, we can use adenovirus, we can use liposomes, and provide the right DNA to overcome the DNA that is. But we are looking for more definite solution that can be very, very targeted. Now you should know, we all know that our human genome is not short. It is 46 chromosome, and it is few feet of DNA, and just 4 bases, ATGC. So it has to be very, very targeted approach in terms of treatment. But what happens that when technology grows, when a technology is discovered, it comes to public domain, then people have ideas, people have concepts, and they want to apply in various model systems, right? For example, mRNA vaccines came for COVID-19. Earlier, it had come for Zika virus, and now they are trying to use this mRNA technology for cancer treatment, to make new type of plants and so on, try in different system. So it sometimes research goes into commercial direction, and where is the stop sign? That's a very valid question. So science, technology, how much it should grow, it's a social debate, right? Should we make babies which should be completely edited by technology? Because technology is there. It's just that it has to reach to that level of competency where scientists are involved, the community is involved because they are at the receiving end, and of course government policy is around. So when I teach these things in this course, many of you might go and do public health or do epidemiology based studies in Columbia University and so on and so forth. So when you have this background, then you understand, you take calculative informed decision. So likewise, gene therapy also, it was, the motivation was to treat human diseases, but it diversified, and because people know their potential, people try to weigh the potential of this technology and try to use in different areas. For example, in generating plants for biofuel, reducing the cost of food, more effective medicines. These days, this medicines that we see on the counter in a pharmacy, they're all going to change very soon because of maybe they will become less effective, or we are consuming a lot because of a bioavailability problem. So the newer medicines will keep coming, and so new model systems, a new mechanism to understand disease process. So everything will be. So it's going to affect medicine, biology, and biotechnology. So something that starts as a simple concept, if it is applied, then it becomes technology. When technology comes into on the bench in the lab, then people try to use it in a different model systems. And that's how, that's a starting point. Look at the cell phone, how it started, right? I got my cell phone in late, 2010 or 12, I got just, 7, 8 years back, and I see the change. And I saw the phones before, like in early 2000 or late 1990s, those cell phone and their capabilities, and now the cell phone that are coming. So, just now, today there was an update on iPhone. So many features they're updating, right? They're updating every month or 2 months or 3 months or quarterly. So yeah, so technology it looks like there is no barrier. So coming back to today's topic, uh, the idea to edit gene is to cure. That's the most novel thing, or a novel as well as noble thing to treat human diseases, especially congenital diseases that is acquired from parents from earlier generation, or during the pregnancy, then there is some error. And remember that, living in New York, we live in a metropolitan climate. There is a lot of metropolitan urban stress that can affect the fetus, psychology of mother, and that stress can be passed on through placenta to the growing baby, and it can cause some kind of manifestation. And stress can always, cause DNA, alterations, and people are looking into that. If not, then they can also alter the epigenetic profile of the baby. So all these things are there, and people are recognizing it. But I want to focus this lecture on only those kids or people where where DNA nucleotides has been changed. So how to correct that, right? So there are three technologies that came that came that is being that came into our knowledge to alter DNA sequence. The first is, it says ZFN, zinc finger nucleases. Right? So this, I'll come to it, what zinc finger nucleases are, but they can go and bind to a specific sequence in the DNA, and they can try to break or edit the DNA. The second is tail nuclease. This also can bind to a specific sequence of the DNA, and they can bring some change. The third and the most effective in today's day is CRISPR-Cas9 system. Right? So these two are still at a lab stage. People are trying to understand or using. But with the advent discovery of CRISPR, maybe around 2012 or 2013, it is being very, very effectively, being very popular and being used worldwide. It has a lot of potential to edit a wrong gene. And so, and it is, as far as strategy wise and dollar wise, this is more cheaper and more precise, more neat than these two above technologies. So I'll just briefly talk about zinc finger and tail and then go into CRISPR. So zinc finger, zinc finger is just a protein domain, mainly alpha helix and beta sheet. And then this binds to zinc atom. So this is a metal binding protein, right? And this here, you can see the 2 cysteine, this metal zinc ion. It's a divalent or bivalent ion. It is bounded to 2 cysteines and histidine. Okay. So this this alpha helix has very specific potential that it can bind to the major groove of the DNA. And what scientists have done, this specific major groove binding protein has been fused to nucleases FOC1. FOC1 is a nuclease. So this alpha helix goes and insert itself in the major groove of the DNA, and then this nucleases goes and makes a double strand break in the DNA. So essentially, this is a molecular scissor where it can cut DNA. So there are two portions to the zinc finger binding gene editing system: DNA binding domain and DNA cleaving domain, FOC1. So why DNA binding domain is important? Because you want to alter a specific sequence. Now this technology is still at the lab level. It hasn't gone to, people are trying to modify stem cells and plant embryos, but it has not taken off because our human genome is so big, and remember they're only made up of four bases. So there are a lot of off-target effects, meaning that in addition to binding to the specific target, they also bind to non-specific target. So it's very difficult at this stage to take it to a therapeutic level. So its benefits are very limited, right, in terms of in the lab and generating mutations in the lab, but may not be tried in a human system. The second system is that makes gene editing is transcription activator like effector nucleases. This is called as TALEN. Now, what is transcription factor? So let's say there are many genes in our cells, right? Not every gene starts making RNA on DNA, right? So this process of transcription is started by RNA polymerase, and this RNA polymerase binds to a specific sequence that is called as promoter, right? Now, there are so many genes in the cell that this RNA polymerase needs help to decide which gene to start transcription. So in eukaryotic cells, there are factors that helps RNA polymerase to bind to a specific promoter, so that a specific gene. So every cells have RNA polymerase, but if a specific gene has to be activated, it needs a transcription factor. This transcription factor also, in addition to this transcription factor, one has one essential feature, and that is called as DNA binding domain. This helps the transcription factors to go and bind to the specific promoter. It has a DNA binding domain. meaning this protein can bind to DNA through one of its portion of the protein. So coming back to TALEN, so these are custom made, these are custom designed domains, and they attach to the nuclease. And this nuclease it can cut the DNA specific site. So again, transcription factor can direct this nucleases to a specific sequence on the DNA, and they can mediate this cut, and these create double strand breaks. Okay, so these were two short topics: zinc finger protein and TALEN, transcription activator nucleases. So now I will try to go to CRISPR. CRISPR stands for clustered, regularly interspersed, interspaced, short palindromic repeats. It's a big name, right? So, when I used to read this name, I used to get, sort of scared that why it is so big name. So, and where does this name come from? Clustered, right? Clustered means they're together as a colony, and regularly. Regularly means they're interspaced at a regular positions. Short palindrome. But what is palindrome? Palindrome is a sequence of DNA that is symmetrically, it can be read from both 5' end and 3' end. For example, GAA TTC, if you're reading from 5' to 3', if you make the double stranded that would run from 5' to 3', it would also read as GAA TDC. Now this CRISPR system also has two portions, but before I go into the detail, let me show you this, share something. This CRISPR system is a big discovery in biology. And it says, so I just copy pasted from Google Images, a seismic shift in gene editing. And as I mentioned to you, gene editing, congenital diseases, genetic disorders is going to happen more because of stress level and pollution or various environmental toxicants. So it's perhaps time to have a better option to treat diseases which occur due to genetic mutations. And these two scientists, two ladies, they got award, Nobel Prize award, the highest award in science, and in 2020. And they started working since 2012 or 11. I don't remember the precise date. You can check. And within 8 to 9 years of the work, they got Nobel Prize. And if you listen to their YouTube videos, they caution people that this is such a powerful technology, but people have to, I think they gave a TED talk. And they underline this, that it has huge utility, and people can get over ambition and they can try to make organisms that could be different. Just now, I was reading somewhere, California, they're trying to make a mutant mosquito, Aegis aegyptis. This mosquito carries Zika virus. So this genetically modified mosquito, most likely through CRISPR, can dominate over those mosquitoes that carry Zika virus. So, okay, so then what happens? The other side of it is that you're removing the natural inhabitants of the earth. What will be the cost? We don't know. So, and it has been anticipated by 2030, CRISPR gene editing ability will raise ethical, concerns about the ripple effect, designer babies, and what it would mean, like as I mentioned to you in the beginning of my lecture. So let's talk a little bit about, CRISPR. So this CRISPR consists of two things. One is Cas9. Cas9 is a nucleases. Now, nuclease is an enzyme that can digest, um, DNA or RNA, but this is very specific. It digests the DNA, and this can also bind to a special RNA that is called as guide RNA, right? So please remember this protein here, Cas9. It has a nuclease activity, mainly targeting DNA. It can bind to RNA, and this RNA is called as guide RNA. What exactly RNA is? An RNA molecule is the expressed molecule of DNA. So if there is an RNA, it means that this RNA is coming from a DNA, which is inside the chromosome, right? So this, when Cas9 CRISPR RNA, they come closer to its target DNA. Target DNA means the DNA that transcribe this RNA. They make DNA RNA heteroduplex. So the orange color is the RNA and dark gray color is DNA. When they come together, RNA DNA heteroduplex, it creates a cut in the DNA, right? And this cut, in the DNA, not in the RNA. And this cut is the point where the therapeutic DNA can be ligated. This capability is not seen in zinc finger protein as well as in talons. So I repeat, this is a CRISPR-Cas9 system that has a guide RNA that guides to a specific sequence in the DNA that has to be edited. This makes the cut here in the DNA, and this cut can further be used to paste healthy DNA for genomic editing to treat disease. Now the question comes, from where this such powerful system comes, right? So interestingly, this system comes from bacteria. And bacteria, we have immunity, right? We have innate immunity. That is like skin, tears, nasal epithelial cell, which is moist, nasal fluid. They all work as a tears, nasal, ear wax, nasal mucus. They all work as an innate immunity. Skin, so they provide, they protect us from pathogens or bugs. T cells and B cells, which are inside our body, they also work for innately, but they're more involved in acquired immunity, right? Adoptive immunity, or So so this is the most interesting aspect, and this is the concept that was developed in a lab for gene editing. So this is a bacterial physiology, bacterial genetics, bacterial immunology, bacterial immune memory. We were talking about vaccines and immune memory. So bacteria also has immune memory, immunological memory, and that system is called as CRISPR-Cas9. Now, before I come to this slide, let me just give you some few concepts. So we know that bacteria has restriction enzymes, right? These enzymes, they can cut at specific DNA, Now you can ask me a question that while this restriction enzyme cuts the DNA, why doesn't this restriction enzyme cut bacterial DNA? Why only, foreign DNA, right? So this question you can ask me. So the answer to that is that these restriction enzyme cannot cut bacterial DNA. They cannot cut bacterial DNA because bacterial DNA is methylated. So this methylated bacterial DNA prevents this restriction enzyme to work on the host DNA. So these restriction enzymes, they can cut the bacteriophage DNA, but it will not be able to cut the host DNA or bacterial DNA, because bacterial DNA is methylated and these enzymes cannot. So this is one of the protection system bacteria has already. The second kind of bacterial protection system it has is CRISPR. So what happens here, that when bacteriophage infects the bacteria, what is bacteriophage? Bacteriophage is bacterial specific virus. So when a new bacteriophage infects the bacteria, there is a protein enzyme Cas2. This is a nuclease, right? It cuts the bacteriophage, and then this DNA is inserted or copied into bacterial genome. So this is a bacterial genome, and in particularly this is a Cas locus, Cas CRISPR locus. So what happened here? This is a bacteria, never met this phage before. The phage infects the bacteria. The bacteria, through Cas9, Cas2 is a bacterial protein, cuts a portion of bacterial DNA and then put inside its chromosome, right? Called as spacers. Now what happens, that when the same virus attacks the bacteria, here this spacer, it generates an RNA. This RNA goes and binds to Cas9. Cas9, as I mentioned, is an RNA binding protein. So this Cas9 and this RNA is called as guide RNA. This Cas9 RNA complex, they go and again recognize this bacteriophage that, hey, you entered again, we need to destroy you. So this spacer that transcribes RNA, this RNA is called as gRNA. How many RNAs you have studied until now in molecular biology if you have taken? mRNA, rRNA, tRNA, non-coding RNA, siRNA or RNAi, and this is the fourth or fifth one or sixth one you are studying, is called as guide RNA, which is part of CRISPR-Cas9 system that gives protection to bacteria from viruses. So bacteria, so in our body we have T cells and B cells, or skin cells, tears, mucous membrane, earwax. They all work as an innate and adaptive immune system, immune protection, immune defense. Bacteria cannot have these things. They are unicellular. So they take a print, right? Fingerprint, but this fingerprint is the DNA or RNA from the bacteriophage, and it put that piece of DNA into its genome. So this becomes part of permanent bacterial chromosome, right? And then this will make RNA. This RNA will serve as a probe, just like a searchlight. And if it gets infected second time with the same bacteriophage, this RNA and bacterial DNA, they will form heteroduplex. But before that, this RNA will work with Cas9 to destroy this. So isn't it very similar to our immune system? So we got infected with COVID-19, we developed some immunity. Maybe it was not enough, but it created immunological memory so that if we get second time, if we get infected, then we can protect ourselves. But what we do, we create vaccines. Vaccines, they come into our body, they create a memory. And here it is this spacer within the bacterial genome. So we create immunological memory, which are present in lymph node or spleen. Bacteria don't have bacteria is unicellular. So they create spacers. So what you see here, red, purple, yellowish brown, green, they are all spacers. They are the fingerprint DNA of various bacteriophages, so that bacteria, when they encounter these bacteriophages, they already have the memory and they can counter right away when the bacteriophage occurs. So think about it, how this bacteria involved. So these DNA, these RNA, they work together with Cas protein. What is Cas protein? Cas protein here in Cas9, they are RNA binding protein as well as nucleases. This is the guide RNA that takes Cas9 to the DNA of bacteriophage, and Cas9 is a nuclease. This is a DNA-based nuclease. So this is a little bit more in detail, but it's same thing. This is a CRISPR-Cas9 locus. These are Cas genes. These are various, spacers, which represents the bacteriophage DNA, interspersed with some repeats, which are palindromic, and then they serve as a memory. Whenever, so if a bacteria can get infected with, let's say, green color, then this will make gRNA together with Cas9, they will destroy this. Similarly, if there is a yellow bacteria, this is from a yellow bacteriophage, they make gRNA, and then together with Cas9 protein, they can blast off this kind of. But this system has been used by scientists to target the Cas9 to specific gene which is having defect. That's where this technology takes the turn, right? When you watch movie, every movie has a turn. Here, the turn is that scientists developed into a therapeutic tool. And here, scientists are treating with CRISPR RNA, and they go and then treat the cells, right? So when I teach Mendelism, Mendel genetics to one of my classes, I talk about sex-linked diseases, and one of the disease is sex-linked disease is color blindness, right? And in which a person, a patient, an individual cannot distinguish color. So there is something wrong with the photoreceptor in that eye, and that error comes from DNA. So a right CRISPR can be injected into the eye photoreceptor cells, and they can repair the DNA by first making a nick and then implanting the healthy DNA. So the gene that is mutated is CEP290, that is critical for photoreception, rod and cone cells that detects light or distinguishes color. So these are the elements. You don't need to know these details, but I thought that if somebody is interested or asks question, then, so this is a Cas9 locus. This is again, it's a repeat of previous slide. This is the genomic DNA. This is the Cas9 system. This is the sgRNA that takes the Cas9 to DNA. And this DNA is nick, and a healthy DNA can be ligated here. This is a poor quality picture, but this was a publication that came out that says how to edit the gene. Right? So one has to find the gene of interest, whether you want to work on P53. P53 is a gene that is mutated in 70% of the cancers. And if the P53 is lost, people assume that cancers will grow very fast. So one can target P53 and correct the error and try to control irregular cell growth. So this is the last slide. There are various approaches here, but the most cheaper and most promising is the CRISPR-Cas9 system. So this is a small topic, short lecture, and I finished this class today. So you have your midterm. I already posted the questions. It will be available to you from 7 a.m. until midnight on 14th of April. And questions are very simple, straightforward. And the starting point is you should review the study guide and go back to slides. And after you have your test, enjoy your spring break and stay safe.
Professor, did you get Tatiana Ross? I came a little late.
I just finished the class. I will take care of that.
Oh, okay.
Professor?
Yes.
The assignment that's due today says it was due at 1:46 p.m. and usually your assignments are due at 11:59.
Yeah, thank you for telling me. Let me fix it. Yes.
Okay.
It is happening with me, and I will.
And there's no class Thursday, right?
No class till Thursday, but I will be here in office hours in case anybody needs any question.
Can you repeat what time the exam is open till?
7 until midnight, just regular.
Okay, thank you.
Professor, there was an email saying that Blackboard might be shut down.
1 second. I can only. May I know which quiz, which assignment you are telling to me?
The assignment that's due today, it is, I'll tell you the exact name. 1 moment.
Assignment.
It's the molecular diagnostics. It's due tonight.
Molecular diagnostics. Okay.
Yes.
So let me first, stop the recording.